Hi, my name is Indrani. I am life sciences, and this is my BlueSky proposal about cellular senescence and how it relates to cancer. I'm trying to address cellular senescence, or the science of aging, because it's something that's one of the greatest mysteries in science. We don't know why cells age necessarily. And it's also really important because cellular aging is responsible for not only why we die and why our cells don't work anymore, but also more specific issues like why our muscle tissues decay, and why our eye tissues decay, and why as we age, our vision is impaired, we have increased risk of loss of muscle mass, and we just can't function as we could at our peak. The reason that I'm also focusing on cancer, because the two are related, is because cancer, as we all know, kills so many people each year, and it's a risk that a lot of us are going to face in our lifetime. In addition, on a more personal note, cancer has affected my family very significantly, so I really wanted to look into it. So I propose to look into a specific gene that affects both cellular senescence and cancer, the P16 gene. It's found on chromosome 9, and there are recent studies that have shown that the P16 gene is responsible for cellular aging in that when it's activated, it creates a protein that causes cell decay. At the same time, other studies have shown that when the P16 gene is inactivated, that increases cellular proliferation, and it also increases cancer and how cells don't die. So essentially, cancer is caused by cells not only metastasizing and proliferating, but also that they will not function as normal cells do and that they'll die early. And that's the opposite of cellular senescence, in which they age and then don't work anymore. So the way that I propose to talk about that is that the regulation of P16 genes and how you can either stop the protein from being made or increasing the amount that it is made can affect either cellular aging or cancer. So I proposed using microRNA, specifically microRNA 24, which is a 20 to 30 nucleotide length piece of RNA that can regulate the activation of the P16 gene. So the way that you would use microRNA is that if you had a copy of it, what it would do is through something called the RNA-induced silencing complex, it could prevent transcription of the P16 gene by attaching to a complementary sequence on it and preventing it from being transcribed and also degrading it. At the same time, if you inhibited the microRNA that's already in your body, stopping the P16 protein from being made, then you can keep the P16 gene from being inhibited and cause it to create more protein and cause it to stop cellular aging. So that's a lot, but essentially what I propose doing is, because having a very active P16 gene suggests that your cells will die and decay more frequently, if you create microRNA and then inject it into localized parts of the body, then you can stop your cells from dying. At the same time, if you create a complementary sequence of microRNA made of DNA that will bind to the microRNA and stop it from stopping your P16 cells, then you can stop your cells from aging and then keep them living continuously. So the risk associated with that, stopping your cells from aging, is the risk of cancer, which is why certain research regarding the P16 gene that has exclusively focused on using transgenes to block P16 completely are a big health risk, because if you completely block the gene, then although your cells won't age anymore, as they didn't in mice that seem to live on forever in that study, the mice were at a significantly higher risk for cancer. But if you use microRNA, which in localized injections would only target certain tissues throughout the body, then it wouldn't have permanent effects that a transgene that would affect the entire genome would. And at the same time, you'd still get a mix of the positive and benefits of regulating the P16 gene. So in LS1a, we did a lot of discussion about cellular signaling, RNA, DNA, and basically how genes are transcribed and then how they're coded into proteins that affect the body. So essentially, what my proposal entails is focusing a lot on the cell signaling aspect as well as the cell gene regulation aspect. So what I focus a lot on is how DNA is transcribed and coded into proteins, and rather than affect the DNA itself, which would be a more permanent solution in what transgenes do. What I propose to do is use microRNA, which has more to do with how it's transcribed into protein and have a less permanent effect while still having the same effect, essentially.